Hello students, this is my 5th video on the series CC4 Unit 4, and in the previous video I was talking about the process of glycolysis, and today we will focus on how this pyruvate will become 2-acetyl-CoA, that is 2-pyruvate will be converted into 2-acetyl-CoA under aerobic condition, and finally this acetyl-CoA will enter the citric acid cycle and will ultimately form carbon dioxide, water molecule and energy molecule. So in the glycolysis, this glycolysis process was taking place within the cytoplasm where glucose had entered inside the cell, and the entire process took place in the cytoplasm, and finally 2 molecules of pyruvate, 2 molecules of ATP, and we got 2 molecules of NADH. That was the final outcome of the glycolysis. Now today we will discuss about the citric acid cycle, which is this one. This is the citric acid cycle. Now the citric acid cycle is also known as the tricarboxylic acid cycle, TCA cycle, or the Krebs cycle. So why these names? The citric acid cycle, these names come from the fact that in the cycle the first product that is formed is a citrate. That's why it is called as the citric acid cycle. It is also called as the tricarboxylic acid cycle or the TCA cycle, abbreviated form, because now here you can see there are 3 carboxyl group. I'll zoom it a little bit. Yes, this one. So here we have 3 carboxyl group, and therefore it is called as the tricarboxylic acid cycle, and it is also called as the Krebs cycle because this was discovered by a scientist named Hans Krebs. Therefore, it is also called as the Krebs cycle. Now, you already know that glucose is a 6-carbon molecule. It will enter through the GLUT receptor. 4, 5, 6, yeah. So, 6-carbon molecule glucose will make 2 pyruvate having 3 carbon each, and it will give 2 ATP and 2 NADH. This was glycolysis that took place in 10 steps. Now these 2 molecules of pyruvate will enter inside the mitochondria. And this pyruvate, after entering inside the matrix of the mitochondria, because this is the outer membrane and this one is the inner membrane, the pyruvate will enter inside the matrix and will bind with coenzyme A. This will bind with pyruvate and will ultimately form 2 molecules of acetyl-CoA. While formation of the 2 acetyl-CoA, it will release 2 molecules of carbon dioxide and 2 molecules of NADH. And this reaction is catalyzed by the enzyme pyruvate dehydrogenase complex. Now this is termed as complex because actually there are 3 enzymes, E1, E2 and E3. Therefore it is called as complex. And the process that is taking place within the mitochondria is condensation. Oxidation. The pyruvate is oxidized into acetyl-CoA with the release of 1 carbon dioxide. Now since there are 2 pyruvate, 2 acetyl-CoA, so 2 carbon dioxide, but for 1 pyruvate, 1 carbon dioxide will be released. Now you know that pyruvate is having 3 carbon molecule. Now here 1 carbon molecule from each of the pyruvate will be released. So this acetyl-CoA will have only 2 carbon molecule. So this is how the acetyl-CoA is formed. Now this acetyl-CoA will enter inside a cycle. called as the TCA cycle or the citric acid cycle, which takes place inside the matrix of the mitochondria. Next, I will be explaining this cycle to you. So let us begin with the TCA cycle. So we start from acetyl-CoA, which was formed by the conversion of pyruvate to acetyl-CoA with the combination of the CoA group. Now, in the first step of TCA cycle or the citric acid cycle, the oxaloacetate will join or will combine with the acetyl-CoA and will form citrate. This is the first step of the citric acid cycle. Now, the name of the citric acid cycle comes from the fact that the first product of the formation of this is citrate. And in this particular reaction, one molecule of water will be taken up with the release of the CoA group, that was previously taken up inside the matrix of the mitochondria, and this particular reaction is catalyzed by citrate synthase. This is the first step. And here condensation is taking place. Now this second step, in the second step of the second reaction has been divided into two parts. In the first part, citrate is converted into cis-aconitate. And this is a reversible reaction, so the arrow will go both ways. Sorry, I have made a mistake. The arrow will go in the both ways. Both of them will be pointing in two different directions. And with the release of one molecule of water, cis-aconate will be formed. Since water molecule has been released, so this is dehydration. This is 2S step. Next one will be 2B step. And both these reactions are catalyzed by aconitase, both 2A and 2B. So in the 2B step, isocitrate will be formed from cis-aconitate. This is also a reversible reaction. I made a mistake in putting the arrow. It will be in the both directions. And there will be release of water. The water will be taken up, and therefore this is hydration. This was step 2B, and this is also catalyzed by aconitase. This was the second step of the TCA cycle. Next comes the third step, in which alpha ketoglutarate will be formed from isocitrate. And this particular reaction is not reversible, so just a single arrow. And the release of one molecule of carbon dioxide. And this step will also release one molecule of NADH, which will provide energy. It acts as an electron carrier or electron donor, mostly electron carriers. So this is oxidative decarboxylation, because one molecule of carbon dioxide is being liberated, and the enzyme that catalyzes this particular reaction is called as isocitrate dehydrogenase. This was the third reaction, or step 3 of TCA cycle. Now the fourth step, formation of succinyl-CoA, or the conversion of succinyl-CoA from alpha ketoglutarate. This is also a non-reversible reaction, therefore a single arrow. in this step, another CoA group will come, coenzyme A group will come and join with the alpha ketoglutarate, thus for forming the succinyl CoA with the release of one molecule of carbon dioxide. Therefore, it is called as oxidative decarboxylation. And this particular reaction, it is also been catalyzed by one particular enzyme called as alpha ketoglutarate dehydrogenase. Complex. So this is alpha ketoglutarate dehydrogenase complex. Why complex? Because there are more than one enzyme in this. And in this particular step, another molecule of NADH is also released. So till now, from step 3 and step 4, we are getting one molecule of NADH. Now comes the fifth step, in which succinyl CoA will be converted into succinate. This particular reaction is reversible, so both way arrow will be given. Particularly this step is very important. Here again this CoA group will be liberated. Here GTP will be formed from GDP, or it can also be an ATP. Like ATP can be formed from ADP. It is either GTP will be formed or ATP will be formed depending upon what it is taking up. If it is taking up GDP, then it will form GTP. If it is taking up ADP, then it will form ATP. This way, it can be either GDP or ATP. And here the thing that is happening is called as substrate level phosphorylation. The enzyme that catalyzes this particular reaction is called as succinyl CoA synthetase. This was the fifth step of TCA cycle. In total, there are 8 steps. Now comes the 6th step, in which fumarate will be formed from succinate. This is also a reversible reaction. Here, one molecule of NADH will be released. Not NADH, FADH2, sorry. Yeah, one molecule of FADH2 will be released. And the process that is taking place is called as dehydrogenation. And the enzyme that catalyzes this particular reaction is called as succinate dehydrogenase. Next is the 7th step. Malate will be formed from fumarate. This reaction is also reversible, and it will take up one molecule of water. Hence it is hydration. And the enzyme that catalyzes this particular reaction is called as fumarase. Next is the last step, that is the 8th step, in which from malate oxaloacetate will be formed. This is also a reversible reaction. This step will give one molecule of NADH. here dehydrogenation is taking place, and the enzyme that catalyzes this particular reaction is called as malate dehydrogenase. So these are the 8 steps of TCA cycle, where we can see that we are getting 3 molecules of NADH in step number 3, step number 4 and step number 8, and we are getting 1 molecule of FADH in step number 6, and we are getting either a GTP or a ATP in step number 5. So in total we are getting 3 NADH, 1 FADH2 and 1 molecule of either GDP or ATP. This is the TCA cycle, or you can say citric acid cycle, and this is also called as the Krebs cycle, after the name of the discoverer, Hans Krebs. So step 3, step 4 and step 8 is giving NADH, step 6 is giving FADH2 and step 5 is giving GTP. So I'm highlighting this part. Step 3, step 4 is giving another NADH, step 5 is giving up either a GTP or a ATP, and step 6 is giving 1 molecule of FADH2, and step 8 is giving 1 molecule of NADH. So this is the outcome of the TCA cycle. So in total there are 8 steps. So we can write down what we are getting. 1 molecule of NADH, this is from step 3. Then we are getting another molecule of NADH from step 4. Then we are getting 1 molecule of ATP or GTP. One molecule of FADH, 1 molecule of NADH, this is what we are getting. So if we write in total, so total number of NADH we are getting is 3. We are getting 1 molecule of ATP or GTP and 1 molecule of NADH. So this is what we are getting from the reaction with 1 acetyl-CoA. Now I had already told you that was from 1 molecule of pyruvate actually, but since from 1 glucose molecule we get 2 molecules of pyruvate, so 2 molecules of pyruvate will be converted into 2 molecules of acetyl-CoA, so it will entirely be, this cycle will go on twice for each molecule of glucose. So into 2 if we do, so finally we get 6 molecule of NADH, 2 molecule of ATP or GTP and 2 molecule of FADH.